Today we will discuss about the DNA. DNA is a type of nucleic acid. So there are the two type of nucleic acid in our chapter in our syllabus. First is called DNA and second one is called RNA. So in these two we will discuss about the DNA. So what is DNA? DNA is also known as deoxyribonucleic acid. What is the meaning of deoxyribonucleic acid? If we are discussing about the separated these terms, then we got the five different conditions. First is called D. D means removal. D means removal. What we remove? We remove the oxygen. From which? From ribose sugar. Ribose sugar is a 5 carbon sugar that is found in both DNA and RNA. So what is the difference between both of them? The major difference is removal of oxygen from the ribose sugar's second position of carbon. From the second position of carbon, the removal of the oxygen, then that pentose sugar, which is called the ribose, become deoxyribose sugar. Deoxyribose sugar plus nucleic, because it is found in nucleus. It's found in the nucleus, so it is considered as nucleic, and it shows the acidic nature. It shows the acidic nature, so that's why it is called deoxyribonucleic acid. DNA is also called polynucleotide chain. Again, the another question is comes in our mind, why it is called the polynucleotide chain? Because when we are talking about the poly, that means many. Nucleotide is a monomeric unit of DNA. We remember it is a monomeric unit and form a chain-like structure. That's why it is called polynucleotide chain. That is the basic structure that we will study the Watson-Crick model. But before that, we will discuss about the basic component of DNA. So, the DNA is formed by the nucleotide. So, we will discuss next to the nucleotide. Now we are talking about the nucleotide. What is nucleotide? Nucleotide is a monomeric unit of DNA. Nucleotide contain the two components which is called the nucleoside and phosphate group. What is nucleoside? Nucleoside is further formed by the two different compounds. One is called nitrogenous base and second one is called the pentose sugar. So what is nitrogenous base? Such compounds who show the basic nature and containing the nitrogen are called nitrogenous base, while the pentose sugar, that means 5 carbon sugar. And these 5 carbon sugar is further categorized by the two types. One is called ribose sugar and second one is called deoxyribose sugar, because when we are talking about the DNA, we have specialized sugar that is called deoxyribose sugar, because if from the second position of the carbon we will remove one oxygen. Now, how many types of nitrogenous base? So there are the two types of nitrogenous base. One is called purine and second one is called pyrimidine. Purine is further divided into two types, adenine and guanine. Both are double cyclic structure. One is 5 carbon, one is pentagon and second one is called hexagon. While when we are talking about the pyrimidine, but related to the DNA, there are only two types of pyrimidine. One is called thymine and second one is called cytosine. These two types of pyrimidine are found in DNA, because we are talking about DNA and it is a unit of monomeric unit of DNA. What is phosphate group? It is H3PO4. When it is combined together, it forms the helix of the DNA. So when we are talking about the structure, it's look like this. One is hexagon and second one is called pentagon. While when we are talking about the basic structure of this is called single hexagon. How these are attached to each other? According to the Chargaff rule, the purine always combined with pyrimidine. The purine always combined with pyrimidine and specialize. A means adenine attached with thymine and cytosine attached with guanine with the hydrogen bond. So we will show the dot dash structure. Two hydrogen bond and three hydrogen bond. This concept is cleared by the Chargaff and it is also known as Chargaff law. Pyridine always combined with pyrimidine and pyrimidine always combined with purine. Adenine combined with thymine and cytosine always combined with guanine. Now we will come to the basic structure of DNA. It was given by the Watson and Crick. This is the basic concept of DNA. DNA is a double helical structure that form anti-parallel strands. What is the meaning of anti-parallel? It means one shows the 5 prime in and second one is called third prime in. Against this, the 5 prime end with the 3 prime in and 3 prime end with 5 prime in. So what is the basic structure? These are called base pair. When they are combined together, they form a pair-like structure and this pair is called base pair because both are basic in nature. As we can see in the structure, we attach with the dotted structure. It means these are hydrogen bond. A attached with T with double hydrogen bond and C attached with G with triple hydrogen bond. And it are attached with the phosphate group. It will attach with the pentose sugar, then it is will attach with the phosphate group. And this structure is called nucleotide. But this is not the complete DNA. DNA is a double helical structure. When these are combined together, same in pattern, then it is called DNA. And it is the double nucleotide combined together by the hydrogen bond. And when these are combined to each other, then a form long chain. This is called DNA. The DNA contains one major groove and one minor groove. And the one complete helix, the length is 3.4 nanometer that contain the 10 base pair. And the width means the distance between both the helix is 2 nanometer. So according to the Watson and Crick, nitrogenous base just like if we take it as A or T, A or G, adenine, guanine, anyone can be taken as. And here it is the single cyclic structure. It may be thymine and cytosine. These are attached with a 5 carbon sugar that is called pentose sugar. According to this, we will starting the counting from this position 1, 2, 3, 4 and 5. Because the fifth carbon is the upper side, so this side will be considered as 5 prime in. And when we are talking about it, here it is the inverted structure of the pentose sugar and it shows the third carbon to the upper side. It shows the third carbon on the upper side, it is called the third prime and its 5 prime in its lower side because its fifth carbon is on the lower side and these are combined together by the hydrogen bonds. If it is A and T, then it contains show the double bond, but when it is C and G, then it is shows the triple bond. When these are combined together, means one another pentose sugar combined with the different, now it is attached with the phosphate group. This phosphate group is show the third prime and 5 prime always. Now first we understand about the difference between the ribose sugar. This is called ribose sugar. It is a 5 carbon. The first one, second one, third one, fourth one and fifth one. It is a 5 carbon sugar. Each carbon contains the H and OH bond. So it is called the ribose sugar. It is found in the RNA. Now we are talking about the DNA sugar. DNA sugar is called deoxyribose sugar. So what are the differences between the first carbon is same but the second carbon is containing only single H in the place of OH. So it means we removed the second position oxygen and now it will known as deoxyribose sugar. So it is the major difference between the DNA and RNA. Now we are talking about the how it's come the 5 prime and 3 prime. Because when we are counting, the fifth carbon is on the upper side and the third carbon is the lower side. So it is called the 5 prime. So according to this, the second pentose sugar, it should be inverted. If it will be inverted, that means the third position carbon on the upper side. So it is show the third prime in, while the lower side is called the 5 prime in. So by which the pentose sugar decide the prime of the DNA. So now we are talking about the bonds in DNA. We can see the 4 types of the bonds which found in the DNA. One is called N-glycosidic bond, second one is called phosphoester bond, third is called phosphodiester bond, and fourth one is the hydrogen bond. The hydrogen bond is found between the bases, nitrogenous bases. When the two bases are combined together, it forms the hydrogen bond. इसको H bond भी कहते हैं. This is also called H bond, and it is known as base pair. This is called H bond. So how the N-glycosidic bond? The N-glycosidic bond formed between the nitrogenous base and pentose sugar. Nitrogenous base and pentose sugar. This bond is found between the nitrogenous base and pentose sugar. Where the phosphoester bond form? The phosphoester bond form pentose sugar plus phosphate group. Then it is called the ester bond. This bond is called ester bond. Ester formed by the phosphate called phosphoester bond. So where the phosphodiester bond form? When the two nucleosides are combined together with the common phosphate group, then this bonding is called phosphoester bond. We remember the whole formation of called phosphodiester bond. Means two pentose sugar plus phosphate plus pentose sugar. This type of formation is called phosphodiester bond. Now these are combined together and form a complete structure of DNA.